Hello everybody, welcome back to another AP Biology video. In the previous video, we focused on the different parts of our stereotypical cells, and today is all about the cell membrane. Remember that the cell membrane is the barrier that separates the inside of our cells from the outside of our cells, and it regulates a lot of things in terms of what's going on inside the cell. So we're going to talk today about its structure and its function. We're going to talk about all the different ways that we can transport molecules from one place to another. And this video focuses primarily on the Biology in Focus Chapter 5 for the most part. We do save a couple of the sections there at the end for a later unit on cell signaling. So without further ado, let's jump into it. So if you can recall from the previous video that the plasma membrane is our separating barrier. So it separates the living cell and its cytosol from the aqueous surroundings in which it finds itself. And one of the important features of our plasma membrane is that it exhibits something called selective permeability. If we say something that is permeable, that means that it is allowing substances to cross. And if we are selective about that permeability, that means that we allow some things to cross, but not everything. Now, the reason that we can pick and choose what type of molecules are able to cross our phospholipid bilayer is because of the structure of our phospholipids. If you recall, our phospholipids are composed of two primary parts. We have a phosphate head, and then we have those fatty acid tails. Now, the phosphate being charged means that it is hydrophilic. It loves water. So it is attracted to the water on either side of our membrane. And the fatty acid tails, being composed largely of hydrocarbons, are hydrophobic. They don't like water. So this creates what we call an amphipathic molecule, where we have both hydrophobic and hydrophilic regions within the same molecule. With this bilayer, essentially all of the heads that are attracted to water will orient themselves so that they are facing those fluids. So we get them on the inside and we get them on the outside of our cell. Now, in between those hydrophilic heads, we have the fatty acids, those hydrophobic tails. They're protected away from the water on the interior of our phospholipid bilayer. And we see that phospholipids will orient themselves in this type of way in many different cases. So before we had actual cell membranes, the phospholipids would form what we call micelles. And if you are familiar at all with micellar water that you may pick up at the drugstore to help clean off different makeup, that is water that essentially contains these essential bubbles of phospholipids. So they orient themselves in a way that protects those hydrophobic tails. This structure allows for our cell membrane to regulate those materials that may be coming in and may need to go out. And that is what we're going to look at today. Now, in our cell membrane, we contain just more than those phospholipids. The phospholipid bilayer is the sea in which a lot of other molecules may float. So we have different materials that we will find inside of this phospholipid bilayer, and all together, combined with proteins and carbohydrates and cholesterol, we form what we call a fluid mosaic model. If you're familiar with a mosaic, you know that a mosaic is a collection of smaller pieces that all together make up a larger picture. And that mosaics are not necessarily symmetrical in nature. So we have different parts of our phospholipid bilayer that contain different things in different concentrations, and these pieces can move and change position throughout this bilayer. We're going to highlight a couple of the major structures that we find in our cell membrane across the next couple of slides. But it is important to note that before we look at the specific parts of our fluid mosaic model, that the different sides of our bilayer have different structures protruding from the different faces. So this creates what we call an asymmetrical arrangement of all the different things within our phospholipid bilayer. And the way that this is built is determined largely by what is produced by the endoplasmic reticulum and packaged and sent by the Golgi apparatus. Remember that these are two important organelles that we discussed in the last video. Hopefully you recall that the endoplasmic reticulum, specifically the rough endoplasmic reticulum, does most of our protein synthesis using the ribosomes that are embedded in its structure. And the Golgi apparatus is useful for applying the different chains of carbohydrates to our proteins so that they can know where to go and they can identify different parts of our cell membrane. Looking at our proteins, it's important to note that the structure of our proteins determines how they're going to fit within the phospholipid bilayer. Recall that because our phospholipid bilayer contains that hydrophobic interior, that it will orient itself with other hydrophobic bits of our proteins. So we'll find the parts of our proteins that like to stay away from water. They're repulsed by water within the membrane. And the parts of our proteins that like water, that are hydrophilic, will be found on the outside of our cell membrane interior. So that allows us to create a whole bunch of different types of proteins that play many different roles in the cell membrane. These include, to highlight two major categories, are integral proteins. So these are proteins that go all the way through. So they have one side outside of the cell, one side inside of the cell, and then that hydrophobic bit on the inside of our cell membrane. Contrast that to our peripheral proteins. Peripheral proteins are bound to the surface, so they only have a portion of their structure anchored into the cell membrane, and then the remaining part of the protein is outside of the membrane on one side of the cell or another. So let's talk selective permeability. What about the cell membrane determines whether or not substances can pass through it? Remember that our bilayer is composed of our hydrophilic heads on our phospholipids on the outside, and then on the inside, we have our hydrophobic fatty acid tails. So the first thing that's going to determine whether or not our molecules can pass through is whether they are polar or nonpolar. So remember that if something is nonpolar, it's hydrophobic. So if we're hydrophobic, we interact well with the hydrophobic nature inside of the bilayer. But if we have a charge, be it that we're polar or ionic, we are hydrophilic. So if we're hydrophilic, we do not interact well with the interior of our phospholipid bilayer. That means that we cannot cross. Now, both these types of molecules are usually relatively small. So the second thing that we want to look for as far as whether or not we can move across the bilayer is our size. If we are relatively small, then we can easily pass across that bilayer. That's because we can kind of fit in between the membranes, the phospholipids in the membrane. So we can cross easily through the bilayer. If we're really large, we cannot pass through those phospholipids. We can't squeeze in the middle. So we are relegated to using other methods to cross the bilayer. So whether that is moving through a protein channel or protein carrier, like we've got here in our transporter, or if we're relying on something like bulk transport. So we're going to look at each of these a little bit closer. We're going to start with our protein that are embedded in the membrane. So we're going to look at channels and we're going to look at carriers. So we're going to start with our transport proteins. Transport proteins allow for the passage of our hydrophilic substances. So all of these things that can't cross the membrane, they can use one of these methods of transport through the membrane. The first type that we're going to look at are what are called channel proteins. Channel proteins create, as the name suggests, a channel. So think like this is a tunnel through our membrane where we can pass nicely through to the other side without interacting with the hydrophobic tails in our membrane. One type of channel protein that we rely on a lot are called aquaporins. So aquaporins, as the name suggests, transport water. So this is one of the ways that water can easily get into and out of cells. But let's say that we have a molecule that cannot pass through a channel protein. They may instead use something called a carrier protein. So carrier proteins involve the binding of molecules that cause conformational or shape changes that allow for that channel to open and allow our molecule to pass through. Now, what's important about both of these types of proteins is that they rely on structure to determine which type of substance it moves. So they need to be what we call complementary in their structure. What that means is that they are specific to the type of molecule. Specific to our molecule, and that they have regions that are opposite usually to allow for the binding. So if we have a shape that looks like this, they'll have a shape that looks like this so that we fit together nice. Okay, so now we're going to get into the methods by which different molecules are able to cross the phospholipid bilayer. All of the molecules that we're able to cross without any necessary help, these are traveling via diffusion. And diffusion is this process or tendency of molecules to spread out evenly into available space. So on either side of a membrane, we have molecules that are moving relatively randomly. And so we look at the direction in which they're overall moving in terms of diffusion. And when we eventually reach something called dynamic equilibrium, that just means that we're equal on both sides and that we're moving equally in either direction. So why do molecules diffuse? Well, it comes down to their concentration gradient. So when we say that something is more concentrated, that means that it has more of a substance on that particular side. And we can compare that to something that is less concentrated. We have less concentration on one side. So let's take a look here at this picture. we have some molecules that have high concentration on one side of our membrane. So we here have high on this side of our membrane, and we can compare that to low. So they're going to travel from the area where they have higher concentration to the area that they have lower concentration. And this process is going to continue until we are equal on both sides. When we're equal on both sides, that means that as many molecules move into as they move out. So we're crossing the bilayer in equal quantities. This is what is known as passive transport. Passive because it does not really require anything on the part of the cell. This happens naturally. No energy, it just happens. Things will dissolve in and they will dissolve out through the process of diffusion. So here we have our first of 3 application questions with this piece. So in terms of diffusion, describe what is likely to happen to the particles in the image below. I'll give you guys some time to record your response on your sheet and then we will discuss. So diffusion tells us that molecules are going to move from an area of higher concentration to an area of lower concentration. So what we should see is that our molecules, in this case being selectively permeable to our membrane, will move from one side where they are at higher concentration to the area or the other side where they are at lower concentration. Now, they will continue to move until they reach equilibrium. At that point, they will continue to move back and forth, but they will be at equal concentrations on both sides. So we shouldn't be able to see a difference. Now, knowing that water is one of the more important molecules that we've come across in AP biology, it should come as no surprise that osmosis is a term specifically used to describe the diffusion of water across a selectively permeable membrane. Now, water behaves in a different way compared to what we're talking about when we're talking about molecules diffusing. So what's important to note, and there are a couple of different ways that we can describe this, but we move from a region of lower solute potential or solute concentration to a region of higher solute concentration. So what we're going to be looking at here is the ratio of water to our molecules. So here on this side, we have more water and less molecules. And on this side, we have less water and more molecules. So water is going to move in the direction where we've got less water compared to our molecules. So if we've got a membrane that has a bilayer that's selectively permeable to this molecule, we'll have an increase in our water on one side and a decrease in our water on the other. Now, this is going to be important as there is some math involved to decide how water is going to flow. And the outcome of these differences in water and the ability for it to travel while other molecules cannot plays into the idea of tonicity. Tonicity is the ability of a surrounding solute to cause a cell to lose or gain water. If that solute cannot cross the membrane, if it cannot diffuse, water will diffuse to even out the concentration. So we have 2 categories aside from what we would consider to be equal. So the first one that we're going to look at is what we have when we have equal ratios of water on either side. and that is our isotonic. So iso means equal. So this is our normal cell. As much water moves in as it does out, so no net movement of water. And this is when we have equal ratios. Okay, equal ratios. In cells where we don't have equal ratios, we're going to see some net movement of our water. So let's say first we've got a hypotonic cell. So hypo and hypo, I want you to think of hippo, okay? Think like you puff up your cheeks like we would see in a hippo. That is what's going to happen to your cell. So water moves into our cell, causing it to swell up, and potentially our cell could burst if we get too much water going inside. Think of if you fill up a water balloon too much and then your water balloon bursts because there's too much water. That's what we get in a hypotonic solution. In a hypertonic solution, so we have hyper and hypo. So in a hypertonic solution, that's where our water is moving out. So we are shriveling up. So all of our water is moving out, and this isn't good for our cells either when they shrivel up. So really, this is the level where we want to be over here in our isotonic solution rather than in either our hypo or hyper solutions, because they cause our cells to not necessarily look the greatest. And we get the greatest effect here coming from our animal cells because they don't have a cell wall. They don't have something keeping their structure set in one specific shape. So here's the math. How do we know which direction water is going to travel? Well, the direction is determined by our water potential. Water potential being here represented by the Greek character psi. So it is determined by the relationship of our solute concentration and our pressure potential. So solute potential, pressure potential. So our solute potential is also referred to our osmotic potential, and it's really what's doing most of the heavy lifting here. So it gets its own equation where we have I being our ionization constant, and that is going to be dependent on how many molecules we break into when we're dissolved into water. So if we stay as one piece, our ionization is 1. This will be most of our non-polar molecules. If we split into 2 pieces, our ionization constant is going to be equal to 2. This is mostly with our ionic molecules. We're going to be dealing with 2 major molecules in our practice problems here. So we'll have sucrose. So sucrose is non-polar, and so it is going to stay as one piece. And then we'll be dealing also with sodium, sodium chloride or salt, and that is an ionic bond. So it'll split into 2 separate pieces, sodium and chlorine. Now the remainder of this gets into, so C is our molar concentration. That's C standing for concentration. Our R is a constant, so this is always going to be equal to the same number. This will be listed on your formula sheet, and that number is 0.0831. And then T represents our temperature, and that is going to be in Kelvin. So most of the time we're going to have to convert that. So if we're taking something from Celsius to Kelvin, so Kelvin is going to equal 273 plus our degrees Celsius. Pressure potential doesn't get nearly as much love. It has to do with whether or not we have a closed or we have an open container. If we have an open container where there's no limit on our pressure potential is going to be 0. Any other time, the cell may exhibit some sort of pressure, but that will be given to you. So we will always be calculating either the solute potential or the water potential based on some values that are given, which leads me into our first practice problem today. Let's say that we have a molar concentration of a sucrose solution that is 0.3 molar. This beaker is sitting on your desk in the open air at 27 degrees Celsius. So I want you to calculate 2 things for me. First, calculate the solute potential. And second, calculate the pressure potential. So using both of these pieces, we're going to do some practice with these equations. And I'll give you guys a moment to write those on your sheet, and I will be back to explain. So the first thing that we can do that's really easy is determine our pressure potential of our solution here. Because our beaker is at open air, that means that our pressure potential is 0. So that one's pretty easy. Solute potential, on the other hand, we need to get into this equation, the negative I CRT. So first off, we have a sucrose solution. Sucrose does not break down in water, so that means that our I in this case equals 1 because it will stay in one piece. So we'll have negative 1, and then our concentration in this case is our molarity of our solution, 0.3 molar. And our R is a given value. R is always going to equal the same thing, so 0.0831, and that is given to you on your formula sheet, so don't worry about memorizing that. And then finally, we come to temperature. For temperature, we're going to need to convert this to Kelvin, so we have 273 plus our temperature at Celsius, 27 degrees Celsius. And this is going to be the initial setup of solving for our solute potential. If I throw all of this into my calculator, I should get a result of around negative 7.479 bars. And so this is my solute potential in this case. So we can describe the movement of water in a few different ways. The first way being that water moves from a high water potential to a low water potential. Now, because our water potential is described in terms of negative bars, this means that we move from areas that are less negative to areas that are more negative. So that means that if we had a -3 outside of our cell and a -2 inside of our cell, that our water would move in the more negative direction. So this is inverse to our other 2 ways of describing. So we move from low osmolarity, so lower concentrations, so low molars to high molars. So if we had 0.3 moles on the outside and say 0.5 moles of a solute on the outside, on the inside, we are going to move into the cell. Now, it's okay to get confused by the different ways that we can describe the movement of our water. What's important is that we have the math to back it up. So we're going to do one more application problem with that equation. For this application problem, I'm giving you two different water potentials, one that is given and one that you have to solve for. So we have a cell, specifically a root tissue cell, and one of the things that I ask you in this application question is to figure out what the solute potential is inside of our cell. And then outside of our cell, I give you that it is negative 4.6 bars. And from that, I want you to figure out which direction is the water going to move in. So similar to the last time, in order to figure out the water potential inside of our cell, you have to figure out our pressure potential and you have to figure out our solute potential, that being negative I CRT. Notice here that this time we are dealing with sodium chloride. So I will give you guys a moment to answer that on your page, and I will be back to explain. Okay, so this question starts very similarly to the previous question where we are just going to be solving for the water potential of our cell, which involves us finding the pressure potential and the solute potential. We still have an open container, which means that our pressure potential is going to be 0. And so therefore, all we have to find in this case is our solute potential, equal again to negative I CRT. So in this case, we are dealing with, oh, where did I put it? There we go. Sodium. Sodium is a metallic compound. So it breaks down into two distinct parts when it gets into water. So that means that in this case, our I is going to be 2. So we have negative 2. We have our concentration of our salt in this case, 0.2 molar. Our constant, 0.0831. And our temperature, which we have to again convert into Kelvin, so 273 plus 20. And if we plug that into our calculator, we should end up with a solute potential equal to negative 9.739 bars. Now, what does this mean? In what direction will our water flow? So if we have inside of our cell is negative 9.739 and the outside of our cell is negative 4.6 given to us up here in our problem, we find that our cell the water is going to move in the direction of the most negative concentration. So our water is going to move into our cell in this case. Okay, let's wrap up the discussion on water and how it moves by talking about plants. Because with their cell walls, the plant cells interact with the movement of water slightly differently than we would see in our animal cells. So they exhibit what we call turgor pressure. Turgor pressure is exerted by the protoplast, so the living part of the cell, the cytoplasm, the vacuole that contains all of this water, and then this plasma membrane on the outside. And it is either going to recede from the cell wall or push against the cell wall. And this helps to keep stiffness in our plant tissues. So if we have a cell that is in a solution that is making it flaccid, its pulled away from the cell wall. So it does not have a lot of water inside of it, so it's kind of limp. Now, if we place it into a higher solute concentration, more water is going to move out of the cell, and it is going to undergo what we call plasmolysis. So plasmolysis is essentially where our cells get really limp, and that is when we have a droopy plant. Now, if we place a regular cell into pure water, so that means that now our water is going to move into the cell. So we have a lower solute concentration on the outside. So we move into that higher solute concentration on the inside, and we become turgid. Our water pushes up against our cell wall and creates a very firm cell structure. So we get a nice, happy plant, a firm plant. Now, if you remember back to the beginning of this discussion, we talked about how some molecules cannot pass through the membrane by themselves. They instead rely on proteins that are embedded within the membrane to move them across. So these molecules don't undergo simple diffusion because they cannot do it naturally. Instead, they rely on help through a process of facilitated diffusion. So to facilitate means to help or aid something along. And so in facilitated diffusion, we have these these protein channels within our membrane that allow molecules to pass through them in order to get from one side to the other. What's important to note with our facilitated diffusion is that we are still moving from a region of higher concentration to a region of lower concentration. So we are still moving with the concentration gradient. Because if we want to move in the other direction, that's going to take a little bit of energy, which gets us to our final big method of transport, and that is active transport. Active transport pushes things against their concentration gradients. So we're moving from areas where we have low concentration to areas where we have high concentration. And doing this requires our cells to give a little bit to help the process along. And so they require energy in order for this to happen. And one of the major processes involving active transport is going to be with our sodium-potassium pumps. So our sodium-potassium pumps work in what we call a co-transport type of manner. So with our sodium-potassium pumps, we have sodium, where we have a higher concentration outside of the cell. So it binds with our protein pump with the addition of some energy, allowing us to push our sodium through, and pushing our sodium through allows us to push some of our potassium into the cell. Now notice that our potassium is going from an area of low concentration to an area of high concentration. So in order for this to happen, we have to have energy. Our cell cannot do it alone. So now that everything is said and done, let's summarize what we've discussed today. So we've got two major ways that our cells can move materials from one side to another. We have passive transport, which does not require any energy. No energy for passive transport. And we can contrast that to active transport, which does require energy. Within passive transport, we've got two major categories. We've got simple diffusion, which can also include osmosis, although osmosis kind of fits somewhere in the middle because that is whether or not water is moving through our membrane or through a transport protein like an aquaporin. And we can contrast that to the diffusion that requires the use of a protein channel. So that being facilitated diffusion because it needs help. Now, all of our passive transport is going from an area of high concentration to an area of low concentration. We are spreading out. But if instead we wanted to go from high to low, we need energy. In order for us to move against our concentration gradient, we need energy, and so we rely on active transport. Now, a lot of what we're going to talk about is going to come back up when we discuss cellular energetics, cellular respiration, photosynthesis, all of those energy generating processes in the next unit. One of these key things that we're going to want to keep in mind is the concept of membrane potential. Membrane potential is the voltage that we have across a membrane, created by differences in the distribution of ions. Most of the time, we are going to be considering hydrogen ions, which have a positive charge. When we have more hydrogen ions on one side, we are more acidic. When we have fewer hydrogen ions on one side, we are more basic. And so the movement of these ions is going to be determined by the combination of two forces, creating what we call an electrochemical gradient. So these are determined by our chemical forces, that is the concentration of the ion. So we have low and we have high. And that's also going to be determined by our electrical force. So whether we have more positive on one side or less positive on another side. So both of these are going to work together. And notice here that our hydrogen ions are moving against their concentration gradient. And so through active transport, we need ATP energy in order for those things to move. In discussing active transport, one of the other pieces that we'll often see play into this is the idea of co-transport. When we say that something is co, like you have co-captains on a team or you are co-parenting, that means that we are acting with or together. In these cases, we have two protein channels, one of which is requiring energy to move and then allowing simple diffusion to move something against into the cell that would normally require energy. So we've got two proteins working together to get materials into the cell. Okay, we've got to the end. So the last piece is what happens when we have molecules that are too large even for our proteins. Well, if we have really large molecules, say something that needs to be broken down within our cell, we can get it into the cell and out of the cell if we're making it inside of the cell through a process known as bulk transport. So bulk probably makes you think of something large, and transport, we are getting it across that membrane one way or another. So this typically happens through the use of vesicles, vesicles being these membrane bubbles that are created within our cell. And we have two major categories of bulk transport, and that's going to be largely determined by whether we're going into the cell or we're going out of the cell. So pay special attention to these prefixes, endo, meaning in, and exo, meaning out, and that will help us determine whether or not we're going into the cell or we're going out of the cell. But essentially, both of these processes work in the same general way. It's just direction that changes. So to get materials into the cell, our cell membrane pinches together and forms a bubble vesicle. So that vesicle can then move into our cell. If we wanted to get our material out of the cell, we could take what is a vesicle inside of the cell. It can fuse with the cell membrane and then open and release its contents out into the cell to do all of the good things that it needs to do. So, I know that was a lot. Lots of different things, a couple of new equations for us, but the idea still stands. We have a cell membrane that is regulating what's coming in and what's going out. And there are a couple of different ways for things to move, whether that's passive transport, active transport, or finally bulk transport. With this, we will wrap up Unit 2. And I will see you guys in Unit 3 when we start talking about how our cells are able to make energy. I'll catch you guys then.